on systems level metabolic modeling of glycan degradation of gut microbes.
Alright, thank you very much. I'm going to talk about something completely different, which is the metabolic modeling of the glycan degradation process of the microbiota. And this review was published just this month in Nature Reviews Microbiology, and it examines the importance of glycans, basically being the main fermentation substrate in the gut. They control the species composition of the microbiota, both in infants and in adults, and are therefore very crucial and important for human health. Now, because different bacterial species can degrade different glycans, they present a non-invasive strategy in which we can control for the species composition in the gut. Now, the problem is that there is currently no way to predict whether a bacterial species can degrade or utilize a certain glycan for growth other than time-consuming individual growth experiments. And we propose the framework of metabolic modeling in order to try and predict these utilization patterns. I listed here a couple of few reviews that describe the various purposes of metabolic modeling, and from basic studies of cellular metabolism like gene essentiality study or carbon source utilization, through more biotechnological applications like engineering production strains or bioremediations, and from the same issue of Nature Reviews Microbiology, they even propose that this framework, the metabolic modeling framework, is now can be used to the study of community systems biology, where multiple organisms are simulated together in a shared metabolic environment. Alright, so to give you a basic one-on-one on what metabolic modeling is, basically you start from the genomic information, you start from the genome sequence, okay, of any bug that you choose. You extract the genes from the genome, then based on sequence similarity to known enzymes in databases, you can construct an initial metabolic network, and in this network the nodes are the metabolites and the edges are the enzymatic reactions. Alright? And recently a group developed a pipeline that does all this process automatically, and for each newly sequenced genome you can reconstruct a species-specific metabolic model. However, still the problem is that all of these metabolic models, they don't have any representation of the glycans. So what we propose and what we sought to do in the last year, recent year, was to take all of the bacterial species with published genomes in the Human Microbiome project and integrate them with a set of several thousands of glycans that are available in a database called K-glycan. And we did this using an algorithm that we developed called glycan degradation, or GLIDE. And what this algorithm does is fairly simple. It takes as an input an enzymatic function or an EC number and the glycan, and it asks whether this enzyme, this EC number, can break this glycan, and if so, then how will the degradation reaction look like? Okay, so I'll give you just one example. So let's take EC number 3 to 115, and we did this by manually mapping this EC number into a set of rules that dictate how will the degradation reaction look like. So these are the rules that were mapped to this EC number. So we can see that it hydrolyzes the glucose alpha 1 to glucose linkage. It's specific for, it's an exoenzyme. It's specific for glycans which are larger than 2, rear polymerization. And it's a non-reducing, specific for the non-reducing end of the glycans, etc., etc. There are a couple of more rules which I won't get into. But if we take, for example, this glycan, so we can see that this alpha-1,2 linkage can be broken by this enzyme, and the products will be one glucose molecule and a smaller glycan. So this is one EC number and one glycan, but like I told you before, we can do this over all of the glycan degradation-related enzymes and several thousand glycans. So I just want to describe the workflow. So we started from 194 glycan degradation EC numbers, 7,000 glycans, and 200 bacterial genomes from the Human Microbiome project. Then, using the GLIDE algorithm, we reconstructed 140,000 glycan degradation reactions. All right. However, these reactions are not species-specific, and also they don't have any subcellular localization. So we can extract this information from the genome. So based on the genome annotation, we can map genes to known EC numbers, and also using the P-sort-B algorithm, we can predict protein subcellular localization from the gene sequence. And taking all of these three together, we now have species-specific and localized glycan degradation reactions. All right. So also from the bacterial genomes, we took advantage of the pipeline that I described before, called the model seed, and we reconstructed 205 metabolic models for these genomes, and both of these two together, it resulted in 205 glycan integrated metabolic models. And the type of questions that we can ask using this glycan is, let's say, where is the extracellular environment, and this is the bacterial cell, and it has this glycan in the extracellular environment, and this is an enzyme, whether it's membrane-bound or secreted, that can break this glycan. And the degradation reaction will look like this. We get two glucose molecules. Glucose is subsequently transported into the cell and into metabolic pathways that eventually into biomass, which results in higher growth rate of the organism. Now we wanted to ask, okay, this is all very nice, but do we have meaningful predictions, accurate predictions of the species utilizing a certain glycan? And we used recently published data by the Gordon lab, where they took two Bacteroides species, and they grew each of them in vitro on a set of independently on every time they changed the glycan. So they used 30 different glycans, and we took Bacteroides ovalis as an example, and here you can see the experimental data. So in greens are all the glycans that this bacterium was able to utilize and to grow experimentally, and in red are the glycans that it wasn't able to utilize. In the square here you can see our predictions. This strip, blue strip, is the glycans that we predicted that this bug is able to utilize, and this square is the glycans which we predict it will not be able to utilize. And when you overlay the experimental results over our predictions, you can clearly see that we have many, many more true predictions than when overlaying the experimental results over random predictions. And this is highly significant. Taking a more global view of glycan utilization of the gut bacteria, we wanted to look at the total number of glycans utilized by different species of the gut microbiota. And here you can see that while the majority of the species can only hydrolyze and utilize a limited amount of glycans, there are some species that are capable of degrading a couple of hundreds and even thousands of glycans. And when we beam these species into their respective phyla, we see a very interesting thing, maybe if not totally unexpected, that Bacteroides species can degrade orders of magnitude more glycans on average than species belonging to different phyla. When we do principal components analysis of the glycan utilization patterns of the different species, we can also see pretty nice separation over the first component between the Bacteroides and the Firmicutes. Finally, when mapping glycans to their biological origin, meaning where they can be found in nature, we can see, if we look only at the utilized glycans, we can see that the majority of them actually come from other bacteria. So bacteria are actually breaking the glycans of themselves, but to some extent also from plants and to animals. But what is interesting here, that if we look at specific biological origins and compare the fraction of utilized glycans out of the total glycans from the same biological origin, we see that only glycans from plants are significantly utilized more than what you would expect. So this means, perhaps like we thought of, that gut bacteria, gut microbes are highly adapted to utilizing plant glycans. And just to, in a few words, talk about our future plans, so we want to examine the glycan utilization patterns of known pre- and probiotics, and based on similarity in the utilization patterns, we hope that when we can predict new ones, new prebiotics, new probiotics, sorry. We also want to reconstruct human individual specific metabolic models of the microbial communities based on 16 sRNA data and examine its association with diet and genetic background. And this also relates to a work that we previously published in Nature of Communication, where we studied the metabolic interaction patterns, competitive and cooperative interactions between pairs of organisms, and this could be extended to communities. And finally, we also want to model the interaction between the microbe and the host, and this is by integrating this work of the microbiota metabolism with models of the host metabolism, of tissue-specific metabolism, by reconstructing tissue-specific human gut metabolic models, which is another work which is currently done in the lab. And finally, I want to thank Rafi Zaretsky, which I don't have this picture because he didn't want to give it to me. He was in charge of most of programming, and my three supervisors, Uri Goffman, which is also here, and Martin Kupiek and Eitan Rupin, and thank you for your attention.
Any questions? Over here.
So, do you think that microbiota is not really responding to diet, but is actually grazing on itself? Is that what you're saying?
I'm not sure I understand the
So
question.
I think what you were saying was that the microbiota has the Bacteroides ovatus, or microbiota, I'm not sure which, has capacity to break down bacterial glycans very efficiently.
Yes, they have the capacity to break bacterial glycans, but I show that they're breaking more plant glycans than you would expect.
Okay.
The reason that they break a lot of bacterial glycans is because there are many, many more bacterial glycans than plant glycans, which is what I wanted to show.
So what do you think is the major substrate of value? Do you think it's bacteria, or do you think it's the diet?
I don't know. It's a good question.
Okay, thank you.
So we just heard a story about the two microbial populations that are completely similar but different in transcriptomics. In your approach is not going to show that difference, isn't it?
Yes, you're right, but there are some methods which can integrate expression, for example, metatranscriptomics into the metabolic models, and this way you can control for the genes that are being expressed. But currently, we don't take this into account.
Smile, take a photograph. All right,